Welcome back, everyone. I'd like to continue our discussion on gene transcription. So what we've done previously is we've described the process of transcription as it occurs in a prokaryotic system. So again, the initial characterization of this process occurred in E. coli. So just like I did with replication, I want to compare and contrast to transcriptional events as they occur off of gene sequences in a eukaryotic system. And as you would expect, just like we saw with replication, is that everything is more complicated in a eukaryotic system. So I just want to point out, some of those differences that occur between the two systems. So the first obvious one, or maybe obvious, is the idea that in a eukaryotic system where you have the genetic material sequestered within the nucleus of the cell, is that the two steps of gene expression, transcription and translation, are going to be uncoupled. So in a eukaryotic system, again, transcriptional events take place within the nucleus of the cell. However, translational events, protein synthesis, which require protein synthesis machinery, which are the ribosomes, those are out in the cytoplasm. So translational events take place out in the cytoplasm. So in a prokaryotic system, that certainly is not the case because you don't have a nuclear envelope. So again, the genetic material is contained within a region of the cytoplasm that's referred to as a nucleoid. And then the ribosomes, again, are present within that cytoplasm as well. So there can be a coupling of both events of gene expression in the prokaryotic system. So in other words, simultaneous transcription and translation occurring. So we'll talk more about that idea when we begin to talk about the second step of gene expression, translation, or protein synthesis in an upcoming lecture. So in addition to that difference is that there's another difference with regard to the nature of the RNA polymerases that are involved in transcribing a gene sequence. So again, what we saw in the prokaryotic system is that there's a single form of RNA polymerase that has the ability to transcribe all three major gene types, the tRNA genes, the rRNA genes, and then the protein encoding genes that produce mRNA. Okay, so a single species of RNA polymerase transcribes all three of those gene types within a prokaryotic system. And what we saw is, again, that can be accomplished by that single form of RNA polymerase because the promoters for those three different gene types are all the same. Again, remember, they consist of the Pribnow box. We talked a little bit about the Pribnow box as a consensus sequence, and then that -35 region as well. Okay, well, it's very different in a eukaryotic system. So in a eukaryotic system, you have multiple species of RNA polymerase. So here what I've indicated is that there's RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. And so each of these different forms of RNA polymerase have the ability to transcribe different gene types. So RNA polymerase 1 will transcribe only ribosomal RNA genes, rRNA genes to produce rRNA. RNA polymerase 3 will transcribe tRNA or transfer RNA genes, creating tRNA transcripts. And then lastly, RNA polymerase 2 will transcribe protein encoding genes that then give rise to mRNA transcripts. So these three different forms of RNA polymerase do have the similar feature as the RNA polymerase that I described in E. coli in the prokaryotic system, in that they are comprised of multiple subunits. Okay, so again, remember for the RNA polymerase from E. coli, there's the alpha subunit, the beta prime, and the sigma subunit. There's others as well. But you get the idea is that they're, multimeric or multi-subunit complexes. So it's the same with these RNA polymerases. in a eukaryotic system as well. And some of those subunits are shared. Again, remember all three of these different forms of RNA polymerase, they're all RNA polymerases. They have the ability to read a DNA template strand to create an RNA molecule. So they must obviously have some similar subunits to give rise to this similar type of activity. But the ability to discriminate between the different gene types occurs because there are also subunit differences between RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. So they're not identical in terms of their overall subunit composition. In addition to that difference is, in terms of being able to recognize the different gene types to transcribe, in other words, how do they discriminate between the different RNA polymerases, discriminate between the three different gene types? Well, the promoter sequences that are associated with these three different gene types in a eukaryotic system are fundamentally different from each other. And again, that was not the case in the prokaryotic system. Remember, the single RNA polymerase could transcribe all three gene types because the promoters had a very similar organization. They all consisted of that Pribnow box and that -35 region. Okay, so I'm not going to describe the organization for the promoters for these three different gene types in a eukaryotic system, but I do want to give you just a little bit of a feel for what the promoters look like for protein encoding genes, those that produce mRNA transcripts that ultimately lead to protein production or protein assembly within the cell. So again, what you see here, again, very similar to the prokaryotic gene organization for the promoters for transcribed genes, is that you have a +1 site here. So that's the transcription initiation site. Okay, that's where transcription will initiate off of this particular gene sequence. And then what you see, all of the information downstream in this diagram, the arrow pointing to the right, all of that information downstream is going to be transcribed to create an RNA transcript. And that, in this case, again, we're talking about protein encoding genes, so an mRNA transcript. Okay, but all of the information upstream of that position is the promoter region, okay, that has the ability to attract the RNA polymerase 2 specifically to that site that will then eventually lead to the initiation of a transcriptional event. So what do we find within that promoter region in terms of consensus sequences? What we find for these protein encoding genes are what is called a -26 region or sequence, or more formally a Goldberg-Hagness box. So that's very much analogous to the Pribnow box, that -10 region that I described for prokaryotic genes. So what you notice is, again, this sequence, this Goldberg-Hagness box, is rich in AT base pairs, just like the Pribnow box. So again, why do you need that? Why do you have that within the promoter just upstream of where transcription actually initiates? Well, again, remember, to initiate transcription, you have to create a transcriptional bubble. You have to denature the DNA, the double-stranded DNA, into a single-stranded form. So again, it's easier to break apart the hydrogen bonds that hold an AT base pair together than a GC base pair. So it's the same sort of reasoning that I described for the presence of the Pribnow box immediately adjacent to the +1 site for prokaryotic genes, transcribed genes. So again, what you can see is that this region here that encompasses that +1 site as well as that Goldberg-Hagness box is what's called a core promoter, because that core promoter is going to be found in front of every protein encoding gene within a eukaryotic genome. So it's invariant, or very little difference from gene to gene. So those are absolute requirements. So it's referred to as a core promoter. However, upstream of that, there can be other additional consensus sequences that are associated with the promoters of the eukaryotic protein encoding genes. So here in this diagram, they just refer to these additional consensus sequences as upstream elements. Actually, they can be downstream as well. So in other words, they can be all around the gene sequence that's ultimately going to be transcribed, and to produce an mRNA transcript that carries information for protein production. And actually what you find is that there's a tremendous amount of variability in terms of the nature of these additional consensus sequences, these upstream elements. There are actually hundreds of different types. So why do you need that much variability in terms of consensus sequences that you would find within promoters of eukaryotic protein encoding genes? The reason for that, again, is that most eukaryotic organisms are multicellular. And all of the cells of the multicellular organism contain the same genetic information. So how do you produce the many different cell types that you find within the tissues and organs of a multicellular organism? You have differential patterns of gene expression. So certain genes will be expressed only in certain cell types and at only certain times of development. So it's this additional regulatory information, these upstream consensus sequences, that confer that type of specificity to a protein encoding gene sequence within a eukaryotic genome, so that you can have a tremendous amount of regulation as to where and when a particular gene is going to be expressed. So then you can create different cell types, a neuron within brain tissue, or muscle cells within muscle tissue, or within skin, or within the liver. Again, all of those cells carry the exact same genetic information, but what makes them different, develop into different cell types, is there's different genes, different subsets of genes that are being expressed in each of those different cell types. And that differential gene regulation pattern is conferred by these upstream elements, these additional consensus sequences that confer temporal and spatial specificity in terms of gene expression. So very important. And again, it's not to say you don't see additional regulatory information with the promoters within prokaryotic gene sequences, in particular for protein encoding genes, but it's not anywhere near the extent that you see for eukaryotic systems. So again, a fundamental distinction between prokaryotic and eukaryotic systems. So here's, again, just comparing three different genes, protein encoding genes that can be expressed within a eukaryotic cell. And it's just a line side by side to give you an idea of the variability in terms of the organization of these upstream elements. So down here to the right of the diagram, you see for each of the three different genes, the core promoter, they're all the same. They've aligned the +1 site. Then again, about 26 base pairs or nucleotides upstream of that is where you have the Goldberg-Hogness box, the rich in A's and T's, AT base pairs. And so they're all the same in terms of their organization there. But beyond that, because these genes require, different patterns of expression in a multicellular organism, you can see all of the additional regulatory information associated with the promoter sequence is variable. So they're all different up in this region here. Okay. So here's a summary of several differences with regard to how transcription occurs in a prokaryotic and a eukaryotic system. So we've talked about some of these already. We've already mentioned the fact that in prokaryotes, transcription and translation are coupled because you don't have sequestering of the genetic information within a nucleus. However, in a eukaryotic system, because you do, genetic material is contained within the nucleus, transcription takes place exclusively within the nucleus of the cell, and then translation occurs exclusively out in the cytoplasm of the cell where the ribosomes reside. Okay, so we've talked about that already. We'll talk about that more in an upcoming lecture. Then there's this second difference that we've talked about already. So again, in a prokaryotic organism, a single species of RNA polymerase transcribes all three major gene types, but in a eukaryotic system, that's not the case. So in a eukaryotic system, each of the gene types have unique promoter organizations, which are then uniquely recognized by the three different major forms of RNA polymerase. Okay, so we've talked about that difference as well. And then lastly, we talked about, the promoter organization, similarities and differences, and really all that we were comparing there. We just looked at, for the eukaryotic system, protein encoding genes, those that produce mRNA transcripts. The promoters that you find for ribosomal RNA genes and tRNA genes, again, are very different from what I've described for the protein encoding genes in the eukaryotic system. Okay, but for the prokaryotic system, again, they were all the promoter organizations were all the same for those three different gene types. Okay, so there's two other differences that I want to discuss just briefly here. So the first one is that what it says is that in bacteria, mRNA, so again, now we're talking specifically about protein encoding genes in the two systems, in a prokaryotic system and a eukaryotic system. So what it says here is that in bacteria, mRNA produced off of these protein encoding genes is polycistronic, whereas in eukaryotes, the mRNA is almost always monocistronic. So what does that mean? What are the definitions of these terms? So polycistronic means that a single molecule, a single species of an mRNA transcript, and carries information for the production of more than one protein, more than one polypeptide. However, a monocistronic mRNA will carry information for the production of only one protein or one polypeptide. So again, a fundamental distinction between the two different systems. And again, what that means is that the gene organization must be different. And we'll talk about that in just a second. So what also happens in a eukaryotic system is then when those mRNAs are produced within the nucleus of the cell, again, remember that mRNA has to find its way out into the cytoplasm to allow for protein assembly. However, prior to that export of the mRNA from the nucleus to the cytoplasm, it undergoes processing. And we'll talk, there's actually three different processing steps that we'll describe. So this processing occurs because of the uncoupling of transcription and translation, or that's one of the reasons. And again, remember, you don't have that uncoupling in a prokaryotic system. You can have transcription and translation occurring simultaneously. So there's no need for processing of the transcript before it can be used for translation. Okay, so I want to explore these last two differences a little bit more closely. So as I said, the difference between a polycistronic messenger RNA and a monocistronic messenger RNA reflects a fundamental gene organization difference between prokaryotic and eukaryotic systems, and specifically organization for protein encoding genes. So let's just go to this diagram here. So here's sort of a generic representation of a protein encoding gene. Here's a promoter, again, whether it's prokaryotic or eukaryotic. Here's a promoter region that attracts, the RNA polymerase to allow for initiation of transcription. Here's the region of the gene that will be transcribed that is informational, right? Say for protein production, carries information for protein assembly. And then lastly, at the other end of the gene, you have information that terminates a transcriptional event. So we talked about termination in a prokaryotic system, the row-dependent and row-dependent forms of termination that take place for genes within a prokaryotic genome. Okay, so that's a generic transcriptional unit or gene sequence, whether you're looking at prokaryotic or eukaryotic. Well, let's look at the details of genes in each different system. And now specifically, what we're looking at are protein encoding genes. So in a prokaryotic system, what it means to be polycistronic is the following. So up here, again, to the left, you have regulatory information or promoter sequence information, referring to this region up here. That allows for transcription to initiate at that +1 site. And so then transcription will proceed through the informational part of the protein encoding gene, and then eventually terminate at a transcription stop site, row dependent or row independent termination. So once that transcript is produced, what you can see is that they've indicated it here as gene 1, gene 2, and gene 3. That single mRNA transcript can carry information to give rise to 3 different proteins that can be structurally and functionally different from each other. Okay. So in any event, that's what we mean by polycistronic. A single messenger RNA can carry information that leads to the production of 3 different types of proteins, all from all of that information being carried within a single mRNA transcript. So the prokaryotic system is different. It's monocistronic. So here again, the idea is that upstream to the left, you have a promoter sequence information. So again, representing generically up here at the top, what I indicated earlier. So then you have a transcription initiation site, the +1 site. So that's indicated here. Then you get a transcript. So once you've initiated, you transcribe through the informational part of the gene. And then down at the other end, you have signals, you have information that terminates a transcriptional event. So there is a difference for eukaryotic genes relative to prokaryotic genes in terms of how termination comes about, termination of transcription comes about. In a prokaryotic system, again, it's very precise. So again, using those mechanisms that I've described previously. However, in a eukaryotic system, termination of transcription is imprecise, meaning that transcription stops within a region of the gene sequence and not at a specific site. Okay, so we'll get back to that idea in a little bit. Okay, so in any event, once you produce that transcript, presumably then that transcript can then be exported to the cytoplasm and be used for protein production. However, that's not the case. As I said, there's a requirement for processing events. One of the processing events that's required is the removal of these sequences within the gene and become part of the transcript that we call introns. So those introns are non-informational in terms of protein production. So therefore, those sequences have to be removed from the initial transcript before the final messenger RNA can be exported to the cytoplasm and actually produce, tell the ribosome to produce a protein, because those introns are non-informational in terms of protein production. It's the exons, these boxed regions that are part of the gene and become part of the transcript, that are informational for protein production. Okay, so what this means is that in the gene organization containing these exon and intron sequences is fundamentally different from the prokaryotic gene. We didn't see introns, non-informational sequences that are transcribed within the gene sequence itself. Okay, and the other thing is that these eukaryotic genes, when in this example, all 3 of these exons are joined together after the removal of the introns, they carry information for the production of a single protein, not more than one. So again, what that means is that they are going to be monocistronic. The final mature messenger RNA product after the removal of those introns is going to carry information for the production of a single protein during translation. So let's look at these. What I had said earlier is that there are 3 processing events that actually take place once an initial transcript is produced off of a protein encoding gene within a eukaryotic nucleus, the nucleus of the cell. So here's the gene sequence up top, double-stranded DNA. It represents a transcriptional unit or a gene sequence. And again, specifically, we're talking about protein encoding genes. So a gene that would be transcribed by RNA polymerase II. So the initial transcript is produced from the +1 site to that region of termination. So again, starting here to a region of termination down here. All of this information is transcribed into what's called an initial transcript or a pre-mRNA transcript. So now that initial pre-mRNA transcript has to be processed. And there's 3 processing steps. So the first processing step is a chemical modification to the 5' end of that transcript. And actually what you do is you add an extra nucleotide. In general, that's referred to as a 5' cap. More specifically, what that cap is it's 7-methyl-guanosine. So it's just a modified form of one of the nucleotides that is normally contained in an mRNA transcript. It's a methylated form of guanosine 5' monophosphate. Okay, so that's the first thing that happens. The question is, well, why does that processing event take place? Why do the others take place as well? So in the first case, it's not 100% determined as to why these processing events take place, but presumably, again, remember this transcript has to be exported through a nuclear membrane out into the cytoplasm. It has to find ribosomes, and then it has to participate in the process of protein synthesis. So that cap can serve multiple roles. It could be involved in conferring stability to the transcript. It can be involved in transport. It can be involved in recognition of the translational machinery. And so it could serve multiple functions or multiple roles. At the very least, it does seem to confer stability. So one of the fundamental differences, and I think I've mentioned this before, based on the chemistry of RNAs and DNAs, is DNA is a relatively stable molecule. It degrades slowly, whereas RNAs are easy to degrade. They fall apart, meaning that they fragment, break phosphodiesterol linkages. They break down into smaller pieces very rapidly and very easily. So any way you can confer stability to the molecule, the better. So it's believed that 5' cap has something to do with conferring stability. Okay, the second step of processing that has to take place is modifications to the 3' end of the transcript. So again, remember I said when termination occurs, transcription termination occurs, it occurs within a region and not at a specific nucleotide. So that means when you produce multiple transcripts off of a single gene sequence, protein encoding gene sequence in a eukaryotic system, is that they're all different at their 3' end. Chemically different. I mean, some of them have more nucleotides than others. So what you have to do is make all of those multiple copies of the transcript identical at their 3' end. So that requires a trimming event. So again, that's what they're trying to indicate in this diagram here up at the top, is that you cut all of the transcripts at their 3' end so that they are then all chemically identical at that 3' end. So a trimming event takes place. So once that trimming occurs, is that there is then subsequently the addition of what we call a poly-A tail. So the addition of adenosine 5' monophosphate residues to the 3' end of that transcript. Okay. So I should point out is that this is true for the 5' end as well as, the modification at the 5' end and the 3' end, is that in both of these instances, what you're doing is you're adding extra nucleotides to an RNA molecule. And you're doing it in a unique manner relative to what we've talked about previously. So again, RNA polymerases require a template. Okay, well, these modifications are occurring in a template-independent manner. So that 5' cap and the poly A tail are added to either end of the transcript, without the assistance of a template strand or template molecule, either of DNA or RNA or whatever. So it's template-independent addition of these modifications to the 5' and 3' end. Okay, so that's the second modification to the initial transcript. Then there's the last one. And the last one we've already referred to. This is the removal of the introns. Again, as I said earlier, the introns do not carry any information that will direct a ribosome in protein production. So non-informational. Only the exons carry that type of information. So the introns have to be removed. They have to be cut out, spliced out. So that's what happens. So once here they're in the example, they're given 2 introns that are cut out. When we say cut out, you break the phosphodiester linkages and then you join the cut ends back together. So you get these 2 fragments, the 2 introns by themselves, and now you've created a messenger RNA from which those introns have been removed. Okay. So the question is, or something you should at least think a little bit about, is how does that happen? Again, remember, how is an intron recognizable as an intron? Again, remember, the overall transcript is just an RNA molecule, the way that we've described them previously. So how does cellular machinery splice out specifically intron sequences only? Well, again, what that means is that there must be consensus sequences associated with the 5' and the 3' end of the intron that allow it to be uniquely recognizable by the splicing machinery in the cell so that they can very precisely be removed. And that's exactly what happens. It's a pretty complicated process. This whole process of removing those introns involves, splicing machinery, which is fairly complex. It involves a complex that's called a spliceosome. Okay, so in any event, after these 3 processing events occur on that initial transcript, that pre-RNA transcript, you now have a 5' cap added to the 5' end. You have a trimming and the addition of a poly A tail to the 3' end of the transcript, and you've removed the introns. So that now mature, what would be described as a mature messenger RNA, can be exported out to the cytoplasm and be utilized by the translational machinery, the ribosome. Okay. So the last thing I should say as well, just to wrap this up, is this modification to the 3' end, similar to the 5' end, presumably it can serve multiple roles, multiple functions. Why do you need to add that poly A tail? Well, presumably it adds some stability to the transcript. It may be involved in transport. It may be involved in recognition by the translational machinery. It may be involved in determining how many times a given transcript is used by a ribosome to produce a protein product. So it can serve multiple roles. Okay, so in any event, once you have a mature transcript like this from a type 2 gene or a protein encoding gene, that it can then be utilized for protein production, for translation. So that's what we're going to explore in our next lecture, talk about some of the details about the translational machinery and the actual process of translation or protein synthesis.